Negative selection is driven most efficiently by bone marrow-derived antigen-presenting cells. Negative selection in the thymus can be mediated by several different cell types. The most important are the bone marrow-derived dendritic cells and macrophages. These are professional antigen-presenting cell types that also activate mature T cells in peripheral lymphoid tissues. The self-antigens presented by these cells are therefore the most important source of potential autoimmune responses, and T cells responding to such self-peptides must be eliminated in the thymus. Experiments using bone marrow chimeric mice have shown clearly the role of thymic macrophages and dendritic cells in negative selection. In these experiments, MHC AXB F1 bone marrow is grafted into one of the parental strains. The MHC AXB T cells developing in the grafted animals are thus exposed to the thymic epithelium of the MHC A host strain. Bone marrow-derived dendritic cells and macrophages will, however, express both MHC A and MHC B. The bone marrow chimeras will tolerate skin grafts from either MHC A or MHC B animals. And from the acceptance of both types of skin grafts, we can infer that the developing T cells are not self-reactive for either of the two MHC antigens. The only cells that could present self-peptide MHC-B complexes to thymocytes and thus induce tolerance to MHC-B are the bone marrow-derived cells. The dendritic cells and macrophages are therefore assumed to have a crucial role in negative selection. In addition, both the thymocytes themselves and the thymic epithelial cells also have the ability to cause the deletion of self-reactive cells. Such reactions may normally be of secondary significance compared to the dominant role of bone marrow-derived cells in negative selection. In patients undergoing bone marrow transplantation from an unrelated donor, however, where all the thymic macrophages and dendritic cells are of donor type, negative selection mediated by thymic epithelial cells can assume a special importance in maintaining tolerance to the recipient's own antigens. And that's negative selection is driven most efficiently by bone marrow-derived antigen-presenting cells. Have a great day.